Hi everyone! This is the first of 3 videos that talk about the various aspects of protein synthesis. And by this time, you should have noticed that this particular unit is very heavy in Skill A, which is models and representations. Everything that we know about DNA and protein synthesis and replication is all based on some very sophisticated investigations in cellular biology and chemistry. In order to understand how all of those investigations come together, we actually use models to show what the thinking is of the people who are interpreting the results. When Watson and Crick determined what the structure of DNA was, that structure actually also indicated to them the ways in which DNA did its 2 jobs, which is replication for one, and second, to hold the information for making proteins. Models and diagrams actually help us all to communicate about what we believe is going on and make it a little bit easier for us to discuss. The diagram you see here is actually demonstrating the entire process of protein synthesis, and by the time we finish all 3 of the videos, you will understand what all of this means. This first diagram shows us transcription, which is a process that happens in the nucleus. To get to the code, helicase unwinds and unzips the double helix. The purpose in transcription is to copy the code for one section of the DNA, called the gene. Because there are letters on both sides of the ladder, one of those sides does not contain the information from the gene. This is called the coding strand. The strand that has the gene is called the transcription strand. Because the DNA can't leave the nucleus, we need to make a copy of the gene. So an enzyme called RNA polymerase is going to land on the open DNA right about here in an area called the initiator region. And what it's going to do is it's going to take these structures, which are floating around the nucleus. These are free nucleotides, and they happen to be free RNA nucleotides because they're going to be making a structure called mRNA. RNA polymerase will be using the DNA as a template as it follows the complementary base pairing rules, putting the RNA nucleotides into the DNA template. As it does that, it is going to be making this structure here, which is the mRNA. It will continue to move down the length of the DNA until it gets to the end of the gene. We can see the RNA polymerase is represented by this large gray circle that's in the back of the DNA. Once the RNA polymerase gets to the end of the gene, the mRNA is complete, and it is going to detach from the DNA template, and it's going to carry the gene out of the nucleus so it can go to the cytoplasm where it can be translated. One of the things that scientists have learned about DNA is that there are 2 different kinds: coding DNA, which actually will make a protein, and non-coding DNA, which have other purposes in the cell. DNA that codes for a gene is generally referred to as an exon. We need the exons to stay in the mRNA because they are part of the actual code for making the protein. In this piece of mRNA, you can see that there are 2 introns, which are code intruders. These are not part of the code for making the protein, and if they stay in, the protein will actually not fold and glob the way it needs to. So an enzyme known as a spliceosome is going to find the intruders, loop them upward, and clip them out so that when they are done, we have our 3 exons, which are the excellent code for making the protein, intact in the mRNA, and that means our protein will be the right shape in order to do its function. Our spliced mRNA needs 2 more things in order to function properly. First of all, a capping enzyme is going to place a small piece on the 5' end of the mRNA, which is going to help the cell to know that that is the start of translation. In addition, there is going to be the addition of a poly A tail to the 3' end, which is going to help to stabilize the mRNA as translation occurs. The final product of transcription is a mature mRNA that has exons with no introns and has a cap and a tail, and it is going to leave the nucleus and head out into the cytoplasm to find a ribosome.